So the title of this talk is Analytical Methods in Proteomics. I will be talking about uses of proteomics for the characterization of proteins and applications in biosimilarity assays. I will divide the talk in two parts. First, we'll talk about an introduction to proteomics, MS and MSMS protein identification, and then we'll discuss proteomic applications for biosimilar comparability assays. Proteomics is the complete study of the structure, localization, post-translational modifications, functions, and interactions of all the proteins expressed by a certain cell, tissue, or organism at a given time under certain conditions. You can perform global or shotgun proteomics experiments where you try to identify all the proteins present in a sample in a certain condition, such as disease development or a specific treatment. Or you can perform targeted proteomics where you try to identify proteins of interest, a group or an individual protein of interest, in a sample in a certain condition. Since proteins and their post-translational modifications can be studied by mass spectrometry through the measurement of their exact masses, the everyday definition for proteomics is the study of proteins through mass spectrometry. To study any analyte by mass spectrometry, we need to go through these three main steps. First, we need to generate gas phase ions of the analyte. Then we need to separate those ions according to their mass to charge ratio, and we need to be able to count or measure the ions. That is why every mass spectrometer must have these three main components. We have the source, where the ions are generated. We have the analyzer, where they will be separated, and we finally have the detector, where they will impact and they will be counted and measured. So there are different kinds of sources, analyzers, and detectors. They can be combined in different ways. You may have more than one of each in one instrument, but every mass spectrometer instrument will have one of these three, one of each of these three. In the case of proteomics, there are two main ionization methods. One of them is MALDI. MALDI stands for Matrix-Assisted Laser Desorption Ionization. For MALDI analysis, the sample is mixed with a matrix, and they co-crystallize on a target plate by the evaporation of the solvent. Then we shoot the laser on the mixture, and the matrix molecules will receive the laser energy, and they will ionize, and they will transfer the energy to the analyte, allowing it to ionize. In proteomics, we usually work in positive mode, so the analytes will be protonized, and since the energy transfer from the laser to the matrix to the analyte is pretty inefficient, the analytes will usually have one charge. They will be charged +1. The other form of ionization we use in proteomics is SC or electrospray ionization. In this case, the peptides are dissolved in liquid phase, and they go through a very fine capillary needle, which is under high voltage, and they vaporize into fine multiply charged droplets. In the case of peptides, we usually will have charges of +2 to +4 or 5, but in the case of big proteins, you may have charges of up to +45 or even more. If we compare MALDI and ESI and take a look at some advantages and disadvantages, we can say that MALDI is a method that is quick and simple. It's high throughput. When I say it's quick and simple, I'm talking about the spectrum generation, not the analysis. The analysis and interpretation of the data is long and complex for any kind of ionization method, but spectral generation is quick and simple. In the case of electrospray, one of the biggest advantages is that since the analyte is in liquid phase, it can be easily coupled to liquid chromatography. This is very important because it allows for the analysis of complex mixtures. The analytes will be separated along the chromatography run, and they will reach the ion source at different times. This ionization method is not high throughput because the runs will take as long as the chromatography run takes, so it's much slower than multi. Then a disadvantage of electrospray is that the sample is consumed once it is injected. It goes through the instrument and it's done, you don't have any more of it. In the case of multi, since it's crystallized on a target plate and this is stable, you can go back to your sample a couple of months after you've loaded it, and you'll still get good spectra. Then for electrospray, we have it's better for low molecular weight analytes, because since there is no matrix, there are no interfering matrix signals. The matrix will ionize and will generate signals in the spectrum that may interfere with your analytes. In the case of multi, we do have the interfering matrix signals, but an advantage is that the spectra are easier to interpret. Why are they easier to interpret? Because we have, can you hear me correctly, or does it go on and off? They are easier to interpret because we have monocharged ions, so we have fewer signals in the spectrum. Now we'll see an example. And we have a direct mass visualization. So here we have two examples, a multi-spectrum on the top, an electrospray spectrum on the bottom. On the x-axis, we have the mass to charge values, m/c from now on. On the y-axis, we have intensity in arbitrary units. So these are spectra for a pure RNase protein. It's around 14 kilodalton in molecular weight. And what we see in the multi-spectrum is a main peak at an m/z of around 14 kilodaltons, which corresponds to the monocharged ion. We also see a much smaller peak at around 7 kilodalton, which corresponds to the doubly charged ion. In the case of the electrospray spectrum, we see a group of peaks. Even though we're talking about an intact pure protein, we get all these peaks that they correspond to the different charges, the different species with different charges of the same protein. So what do we have to do to find out the molecular mass of a compound in a spectrum like this one? We need to deconvolute the spectra, that is, we need to clear all the noise signals, and then we need to multiply the m/z value by the charge of the peak, and we have to subtract the protons that were added during the protonations. How do we find out the charge of a peak? For this, we use the isotope distribution patterns. When you zoom in on a peak like this one that we see over here, which is 685.3, what you actually have is a group of peaks. And why do we have a group of peaks? Because, as you all know, in nature we have groups of natural isotopes for each atom. For example, in the case of hydrogen, we have that 99.985 of the atoms will have a mass of 1 dalton, but there's a minimum percentage, 0.015, that will have 2 daltons. So the first signal in a group of this isotopic distribution spectrum will correspond to the monoisotopic ion, that is to say, the peptide that is made up of monoisotopic atoms, of the most abundant atoms of each kind. But then the following peak in the group will have all the atoms that are the most abundant, but one of them will be a different isotope. So we expect the mass difference between the monoisotopic peak and the following peak to be 1. But if we are taking a look at a spectrum, at a peak that is multiply charged, the m/z of this one difference in mass will be 1 divided by the charge. So if the charge is 2, the difference between 2 consecutive peaks will be 0.5. If it's 3, it's going to be 1 divided by 3, 0.33. If it's 4, 1 divided by 4, 0.25, etc. So if we take a look at these 2 peaks, we see that one of them is, you don't see it, but trust me, it's 685.57, and the next one is 685 0.8, so it's 0.3, so the charge is +3. Fortunately, there are algorithms that will calculate these the charges of the peak, so we don't have to do it manually. And there are also, you can do it with, in the case of very complex mixtures where you have a lot of charges, you can do it with plotting two simultaneous equations. Is with two unknowns, you can determine the charge of the peaks. So in our lab at the CQVM, the mass spectrometry facility I work at, we have two instruments. We have a multi-TOF, which is an ultraflex 2 from Bruker Daltonics. So we have the ion source, it's with the laser, it's a multi. Then we have the analyzer, which is a TOF, time of flight. It's a very long tube where the ions will be separated according to their mass to charge ratios, and then they will hit on the detector where they will be measured, and their time of flight will be calculated. The other instrument we have is a Q-Exactive from Thermo Scientific. It's got an electrospray source. It's coupled to a nano HPLC. We have a couple of lenses that focus the ions. Then we have a quadruple that will also focus the ions, and in some cases will filter ions out. We will then see why this is important. We have a C-trap where they are cooled down, and then we have the Orbitrap mass analyzer. When we want to fragment ions, as I was saying, the quadruple will filter out all those that we're not interested in. They will go into the collision cell where they will be fragmented, and then those fragments will be measured in the Orbitrap analyzer. So now that we know what the instruments look like and what the different parts are for, we can try to understand how we go about protein identification by mass spectrometry. There are two main kinds of approaches. There are MS experiments where what we measure is the exact mass of peptides derived from a protein, or MS-MS experiments, which are also called MS2 or tandem mass spectrometry, where we measure the exact mass of the fragments obtained upon the fragmentation of a peptide. So the first one is MS classical approach for protein identification, also known as peptide mass fingerprinting. What we do here is we have a protein of interest or a group of proteins of interest. We digest the proteins with a site-specific protease, that is to say, a protease that has specific sites where it will cleave. The most commonly used enzyme for proteomics is trypsin. Trypsin will cut at the C-terminus of arginine and lysine, and it's used because it's very efficient, because it generates peptides which have a basic C-terminal end, and because the length of the amino acids it generates is usually very appropriate for the mass ranges in which the instruments work. So we digest the protein, we generate a soup of peptides, and those peptides will go into the mass spectrometer and we'll get the MS spectrum. In an MS spectrum, we have M over Z over here, and each peak corresponds to the mass to charge value of a peptide. So each one of these peaks corresponds to the M over Z of a peptide. So how do I identify a protein with this? We don't. We have software that will do it, and what the software will do is it will digest a database of known sequences theoretically. So we have to select a database that resembles our sample as much as possible. So if we're working with a pure protein, we will use the sequence of the protein. If we're working with a protein extract from a certain tissue or cells, we will use the proteome of that species. We have to tell the software the enzyme that we used. So basically it will cut all the sequences at the sites where the enzyme cuts. And it will generate a theoretical peptide mass list. And with the theoretical peptide mass list and the experimental peptide mass list, what it will do is it will compare and look for mass matches, and it will generate a list of probable protein hits. So an example of one of the search engines is Mascot. It's one of the mostly used softwares for database searching. It's free and it's easy to use. So these are the things that Mascot asks for. It will first ask for a database, so where will it retrieve the sequences from? We usually use NCBI, which is less redundant than SwissProt, but you can choose. You have to tell Mascot which enzyme you used. You have to tell Mascot how many miscleavages you are expecting. That is to say, how many sites you expect the enzyme to skip. This is usually an empirical data that you will take from previous experiments or from the digestion of standards. We usually use 1 or 2 for trypsin because it's pretty efficient. If you're using, I didn't mention that apart from trypsin, there are many other enzymes that you can use when you need them. They are usually not as efficient, so you will probably allow for more missed cleavages with other enzymes. Then you have to choose a taxonomy. As I was telling before, it has to resemble your sample as much as possible. So here you have a drop-down menu from which you can choose different categories. It will probably, it goes up down to the species level in many cases, but in others it doesn't. So for example, if you're working with a human sample, you will be able to choose Homo sapiens from the list, but if you're working with a bovine sample, there's no Bostaurus in it, so you'll have to choose mammals. But you'll go down as, let's say, as low as possible in the categories to resemble your sample as much as possible. This is very important because if your database is too big, the scores of the hits you get will be too low, and maybe they won't be significant. But if your database is too small, you might get that what you're looking for is not in the database, so you won't find it. So it's very important to be as accurate as possible when selecting all these parameters. You have to tell Mascot which fixed modifications you're expecting. That means that if you expect any amino acids to be constantly modified with a certain modification, it has to know it because, for example, in our case, before we digest proteins, we reduce them with DDT to reduce the disulfide bridges, and then we alkylate with iodoacetamide. So we expect every cysteine to be modified with the alkylation. And this generates a delta of 57 on each peptide. So if I tell Mascot, I expect fixed carbaminomethylation of cysteines for every peptide that contains a cysteine, it will add the 57 Daltons of the alkylation. Then we have to specify the variable modifications. Those are modifications that may or may not be present. For example, the oxidation of methionines, which occurs in samples that are stored in protein samples that are stored in a test tube. So whenever it sees a methionine, it will generate actually 2 peptide masses, one with the oxidized methionine, so it's going to have a plus 16, and one without the oxidized methionine. So it's important to regulate the amount of variable modifications you try to search for, because this again will also increase the size of your database considerably. You have to allow for a peptide tolerance, that is, what is the error allowed for the matching of the theoretical masses and the experimental masses, and this is also pretty empirical because you will get it from the measurements of your instrument. You calibrate your instrument every day before you measure, so you know the difference between the theoretical expected masses and the observed masses. You also have to tell Mascot if you're working in positive or negative mode, if the masses you're giving it are monoisotopic or average. Then you can upload your experimental data, or you can just paste the list of masses here, and you perform the search. This is what the result page in Mascot looks like. So it's a probability-based scoring, and significant scores are those who have a p-value that is lower than 0.05. That means that the probability that the hit appears randomly is less than 5%. So Mascot generates this kind of graph where we have the scores on the x-axis and the number of hits on the y-axis, and it shades in green all the non-significant scores. So here it says protein scores greater than 55 are significant, so we see greater than 55 significant scores. We have this over here, And then down here we get the list of hits. We have an accession number, the mass, the score, and the name of the protein. So these are all expected hits. When you click on the accession number, you get the list of observed masses that matched the theoretical masses from the digestion of the database, and these masses correspond to sequences from the database. All this information is theoretical, so the only experimental thing we have over here is the observed masses that match these experimental masses with these theoretical masses with this allowed error tolerance. And then based on all the peptides that we're assuming that we are seeing, we can generate a sequence coverage map. That is to say, in the theoretical sequence of the protein, we color the observed peptides and we get a sequence coverage percentage. So this is how we identify proteins by peptide mass fingerprinting, or MS. There are two important things that I'd like to point out, and that is that although all the peptides in one protein are present in equimolar amounts, that is to say, I digest a protein, I have 5 peptides, and they're all in the same, they all have the same abundance. When you take a look at an MS spectra, you would expect to see this, right? Intensity is proportional to the abundance of a peptide. But what we actually get is this. And why do we get this? Even if the 5 peptides of the proteins are in the same relative abundance, what happens is that different peptides will have different ionization capabilities. Some of them will ionize better, others will ionize not so well. So you will see that they have different intensities. So the intensity will be relative to their abundance, but it will be affected by their ionization capabilities. For example, it is known that peptides with an arginine in the C-terminus will give a stronger signal than those with a lysine, because arginine has a stronger positive signal and better ionization capabilities. And another thing is that the number of peptides identified for a protein depend on the mass of the protein you are analyzing. If I load 1 microgram of a protein, I may see these 5 peptides, but if I put twice as much, I will see the different intensity pattern, but I'll see that all the peaks are higher, and that I get new peaks that corresponds to peptides that were below the detection threshold at this level, but now they can be detected. Another thing, sequence coverage does not have to be 100%. You do need to have more than one peptide mass to identify a protein, usually more than 4 or 5. But you don't need 100% sequence coverage. In some cases, when people specifically want to get 100% coverage, what you can do is to do multiple enzyme mapping, and this means that you will digest a protein with different enzymes in parallel assays. And what do we gain by doing this is that, for example, if we take this pink peptide over here that is generated with a trypsin digestion, we may get that this peptide does not ionize well, or it's not detected because it's too small. And if instead we digest with glu-C, we see that this area is now in this green peptide, which is different and will have different ionization capabilities. So I may be able to detect this area of the sequence that I wouldn't detect with trypsin. So the different enzymes can give complementary information to increase the sequence coverage. Now we move on to the second approach, which is tandem mass spectrometry, or MSMS protein identification. In this case, the first part of the experimental procedure is just like the one we saw for MS identification. But now, once we have the MS spectrum, what we will do is we will select an ion of interest and we will isolate the ion. So in the ion source, we get that all the ions are generated, but then we measure the MS, we select only one ion of interest, and that ion will go into a fragmentation cell. We usually use CID for fragmentation of peptides, that is collision-induced dissociation. So the peptide will fragment by collision with molecules of the same peptide, or you might inject a gas to promote the fragmentation. So you fragment the ion, and then now what we will measure in the analyzer are the ion fragments corresponding to that peptide that you selected for fragmentation. In the case of, so this is a MS-MS spectrum where now each peak corresponds to the M/Z of a fragment of the parent peptide that you selected for fragmentation. In the case of MALDI, the selection of the ions is manual. You will choose an ion that has high intensity and that is separated from their neighboring ions. In the case of an instrument like the Q-Exactive, what you do is you set up a method where you tell the instrument to select the 15 or 10 most intense MS peaks in each MS run for fragmentation. So at the end of a 2-hour run, you may have up to 60,000 MS-MS spectra that will be used for protein identification. If we use CID fragmentation, the peptides will usually break at the peptide bond, and depending on which end of the peptide retains the charge, we will call these ions Y ions or B ions. So for example, for this tripeptide, what we will have is, if the charge remains on the C terminus, we get, and it was broken here, we get, this would be the Y1 ion. If it's broken here and the charge is still on the C terminus, this is the Y2 ion, and this would be the Y3 ion. And the opposite for the B ions. If the charge is retained on the N terminal and it cuts here, we get a B1 ion, then the B2 ion will contain these 2 amino acids. So if we can fragment enough molecules of the peptide to get a complete Y and B series, what we can do is that if we calculate the difference in mass between 2 consecutive ions, for example, between this is an imaginary peptide, and these are all the fragments. Y1 corresponds to this, Y2, these 2, Y3, these 3. So the difference in mass between Y2 and Y3 will correspond to the mass of this amino acid. So by having this delta in the mass, you can predict which is the amino acid that was present in this part of the sequence. So here we have an example in a real MS-MS spectrum, and this is pretty difficult to do. You need a lot of experience and a lot of tips and tricks, but basically once you start identifying the Y or B ions and you calculate the delta between the peaks, you can predict the amino acid sequence of the peptide you fragmented. But fortunately, this is called de novo sequencing, and we don't have to do it on a daily basis because we have software that does this automatically. But if you're working with organisms that haven't been sequenced, this is what you have to do. So what will the software do? So this part, we've already seen it. Once we have the MS, we select a certain peptide, we fragment it, and we have the MS-MS spectrum where each peak corresponds to the M/Z of the fragments of the parent peptide we selected. And what the software will do is it will digest the database, and then it will theoretically fragment those peptides in the database that have the same mass than the peptide we fragmented. So it will then compare the theoretical fragment peak lists with the obtained experimental spectrum. So to summarize, MS identification is based on the match between the experimental masses of the proteolytical peptides derived from a digested protein with the masses of the virtual peptides generated by theoretical digestion with the same enzyme that we used experimentally of all the sequences contained in a database. MS-MS fragmentation is based on the comparison of the experimental fragmentation pattern of a selected tryptic peptide with the theoretical fragmentation of all the peptides of the same M/Z in the database. So both approaches are based on different criteria, and to identify a protein with an MS approach, you need several peptides matching the experimental M/Zs, and to identify a protein with an MS-MS approach, a single spectra with a complete Y and B series matching a unique peptide may be enough. So now we'll talk about we'll talk about biosimilarity assays. A biosimilar is a therapeutical product which is highly similar to the reference biologic that has already been approved for use in patients, and it cannot have any clinically meaningful difference in terms of safety or efficacy from the reference biologic. They are therapeutical compounds similar to human proteins. They are formed by hundreds of amino acids. They will have a three-dimensional structure that will affect their activity, and they will be heterogeneous. They will have high diversity in post-translational modifications. Their origin may be biological, that is to say they are extracted or purified from microorganisms or animals or plants, or biotechnological. They are proteins obtained from genetically modified cells or organisms. We insert the DNA coding sequence and you express the protein. So for all these reasons, they are difficult and expensive to characterize and replicate. There are technical requirements for biosimilarity assays. There's a guide called the ICH Q6B guide, which states that there are 4 main categories that have to be analyzed in order for a biosimilar to comply with the biosimilarity requirements. So there's a physicochemical characterization, which has 2 parts, structural characterization and physicochemical properties. Then we have analysis of purity and contaminants, and analysis of immunochemical properties and biological activities. We will be very quickly going over the categories that can be tackled by mass spectrometry. So we'll see the analysis of primary sequence and disulfide bridges. We'll talk a little bit about post-translational modifications. We won't go into glycosylation, although it can be tackled by mass spectrometry, because you will be seeing it this afternoon with Dr. Alicia Couto. We'll talk about molecular weight and some product-related impurities. So for the physicochemical characterization, primary structure or sequence was traditionally performed by Edman and terminal sequence, which is slow, expensive, and very labor-intensive. Nowadays, it can be done by mass spectrometry in a pretty simple and easy way. So this is an example from an application note from Waters, where the first thing we can do is to do a liquid chromatography profile comparison of the intact protein. So there's no digestion here. We just analyze the intact protein by liquid chromatography. This is a C4 reverse phase column. And we can see, if we compare the innovator with the biosimilar, you can see at a first glance that these 2 peaks are the same, but here we are having some differences. So then what we do is we do mass spectrometry on these peaks, and this corresponds to this first peak over here. And you can see that the raw MS spectra and the deconvoluted MS spectra, they look the same for the innovator and the biosimilar. But if we look at the deconvoluted spectra for this peak over here, we can see at a first glance that there are differences. Here we have a peak in the biosimilar that's not present in the innovator, and the opposite over here. So just by doing liquid chromatography profile comparison, you can tell that there are some differences. Then the next step is to do tryptic digest LC profile comparison. So now it's not the intact protein, but you have a digested protein. In this case, it's with trypsin. So we do a mirror plot of the innovator and the biosimilar. We already know what each peak corresponds to in the innovator, so all the peaks are labeled. So this corresponds to a light chain of trypsin peptide 17 and whatever. So we can compare the mirror plots and check to see if there are differences between the two LC profiles. Then the next step is, so we did the digestion, we separated the peptides and we compared the profiles, and you can do MS analysis of all those peaks that we just saw on the chromatogram. So when you do the MS analysis, what you will get is an observed mass for each of those peptides, and you can compare them with the theoretical expected mass in the innovator. So these are tables that already exist for the innovators, and so all you have to do is compare your sample, and you will usually run an innovator at the same time in case it's a problem with your instrument, and you will compare the masses of the peptides. This is the way to plot the MS analysis. So you have the sequence, and in red you underline all the peptides you saw for each of the compounds. The next step is to do an MSMS, amino acid sequence verification. So what you do is you fragment each of those MS peptides and you compare the MSMS spectra for each one of the peptides you fragmented. So this is an example for trastuzumab, which is a monoclonal antibody, and this we have the LC profile comparison of the intact protein, and you can see that there is a shift in the mass between the innovator and the biosimilar. The biosimilar seems to be shifted in mass. So if we compare the tryptic digest profile, we see you don't see it here, but there are some differences at around this area. And when they do the MS analysis of all these peaks, what they find is that you clearly see there are peaks in the innovator, like this one over here, that are not in the biosimilar, and we do have this peak in the biosimilar that's not present over here. So basically, to check if this corresponds to a difference in primary sequence, what you do is you fragment the peaks, and here we have an example of the two fragmentations, and you can see that the Y1 are the same, Y2 they are the same, but then as from Y3 there is a difference in mass. So here we have the lysine, the threonine, and then here we start with point mutations in the primary sequence. So it's, I wouldn't say very fast, but it's pretty straightforward to do this kind of primary sequence analysis with mass spectrometry. Then an example on disulfide bridges. It was usually done by Elman assay for 3 thiols, but now it can be done by comparing intact and reduced and alkylated protein. So what we do is you have, this is an example of a monoclonal antibody. As you can see, it has a large number and intercrossing of disulfide bonds. So basically you can digest this protein with an enzyme. In this case it's lyse-C, it cuts at the C terminus of lysine. You can get an aliquot and you reduce it with DDT, and then a different aliquot you do not reduce it with DDT. So you basically compare the peptides obtained from the native digested protein and the reduced digested protein. This is a simplified schematic diagram of the antibody we saw on the previous slide. And we have all the expected peptides that are generated with lysine, and all the disulfide bonds are shown. And we can see how, for example, in the case of this tripeptide over here, how they are bound by the disulfide bridges. So we have this H9, H10 peptide bound to this H7 peptide, and bound by this bond over here to the L13 peptide. So if you generate a table with all the expected theoretical masses for the native protein, these are the masses of the fused peptide. So this is a tripeptide we were talking about with a mass of around 5 kilodaltons. And then these are the masses expected for the reduced protein. So now the components of the tripeptide will be separated. We have the H7, L13, H9, first 10 with their corresponding masses. So if we analyze this by mass spectrometry, this is the non-reduced digested protein, and we get the expected peak of the tripeptide at around 5 kilodaltons. But then in the reduced protein, what we see is that this peptide is now very, very small. There are some remnants of the tripeptide, but it's very low intensity. And what we do get are peaks corresponding to the now reduced peptides. So we have the H7, the H9, H10, the L13, you don't see it because it's too small, it's out of range. And then we also have some remaining D peptides that were not completely reduced by the DDT. But if you do this for every expected disulfide bridge, you can predict all the number and the sites where the disulfide bridges are. You can even go on and fragment those tripeptides to check their sequence, and the only thing you have to pay special attention to is that in the Y series, for example, in this case, we will have Y1, Y2, Y3, but as from Y4, we expect the masses of the fragments to be altered with the fused peptide that it has. So as you can see in this area of the spectrum, all these ions have this crossover here, which corresponds to the delta in mass of the 8,9, 8,10 peptide. So it's 1605. So all these fragments that we see here, they have this extra mass of the fused peptide. Then molecular weight, we have seen it in the beginning, but it's pretty simple. You can do it by size exclusion chromatography, SDS page, but with an MS spectrum, it's very easy to see, because basically in a multi-spectrum, you'll expect to see the peak of the expected M over Z, or in an SE spectrum, you'll deconvolute it and get the molecular weight of the compound. And this is also useful, the MS spectrum, for the analysis of impurities. If we are looking for product-related impurities, such as molecular variants of the desired product, the most common molecular variants could be, for example, you may have precursors, unprocessed or uncleaved proteins that are still present in your sample. You can check in an MS spectrum, you will see them very easily. You will be seeing peaks that correspond to masses that are not the expected ones. The same for truncated or aggregated forms. You may have, it's very common to have N or C terminal cleavage or internal cleavages, or you may have dimers or presence of unwanted monomers. So there are different techniques to see this, but an MS spectrum will be fast and simple. You can also look for amino acid substitutions. It's very common to have point mutations, or to have norleucine incorporated instead of methionines, or norvalins instead of leucines when working with E. coli, with what Dr. Burgess explained this morning about the transfer RNAs. It's common to have adducts, glutathione or cysteine bonds that will generate a delta in the mass of the peptide, which is easy to see by mass spectrometry too. Then we may have lysine carbamylation. If we're working with urea, it's very common to get lysine carbamylation, which generates a delta of 43. So again, we do a spectra, we see it. Deamidation of asparagine to aspartic acid can also be seen by mass spectrometry. It can also be done by peptide mapping, because you get this enzyme, for example, ASP-N cuts at the N terminal of aspartic acid. So you can, by digesting with this enzyme, if it cuts or if it doesn't cut, it will tell you if you have an aspartic acid or an asparagine. Pyroglutamate formation is also very common, and you can distinguish the change in mass. And then post-translational modifications can also be detected by a simple MS spectrum. So we can see acetylation with a delta of 42, oxidation of methionines plus 16, phosphorylation, you'll see a plus 80. It's important here to clarify that if you want to focus your study on PTM analysis, what you will have to do is to perform an enrichment of the modified peptides that you're interested in studying, because the post-translational modifications are usually low abundant and not very stable inside a mass spectrometer, so it's important to enrich your sample in the modification you're interested in studying. And then you may need to use different fragmentation strategies, such as ETD, for example, which is softer on phosphorylation. And you may have to complement your studies with enzyme treatments, such as glycosylases or phosphatases, to get more information on those PTMs. But if you're doing a contaminant analysis, for stable PTMs, you can see them in a simple MS or MSMS spectrum. A couple of examples. So this is an example of a deamidation for a certain peptide, and if we compare the innovator with a biosimilar, we can see that the series are the same at the beginning, 175, 276, 439, but then as from this Y5, we have this is 600, 610, 611, so there's a 1 M/C difference in between the two that corresponds to the deamidation of the asparagine. And then we have another example of an acetylation. This is the main peak in the MS spectrum, and then we can see that for the biosimilar we get a shift in 42, so it's very straightforward to see the acetylation here, and you can verify it by the MSMS spectrum, but only just this would be enough to say that you have an acetylation in your sample. Finally, to close up, I'd like to mention the services that we perform at the mass spec facility at the University of Buenos Aires. We do identification of pure proteins and also identification in semi-complex and complex mixtures. We do co-immunoprecipitation and pull-down protein identifications. We do label-free quantitation of relative protein abundance in different samples, intact mass protein determination, and we perform a battery of biosimilarity assays consisting on protein molecular weight determination, complete primary sequence coverage with multiple enzyme digestion, disulfide bridges, number and mapping, point mutation analysis, and stable PTM analysis. This is our website and this is our email in case you want to contact us with questions or whatever you may need. Thank you.